I'll tell you about the different types of grafts or transplantations that are done to describe what is the basis of acceptance or rejection of a organ graft. The methodologies we use to determine whether a graft has a higher chance of being accepted or rejected. And then finally, just to revise what you learned in the hypersensitivity reactions last year in terms of the mechanisms that are involved in graft rejection. So first of all, a bit of nomenclature. So there are different ways of engraftment. The one is in the which a common form is people who suffer from multiple, from severe burns. What they do is they'll take bits of skin from other parts of the body and then graft it on. What they're doing now also is they're able to culture skin cells in the lab into sheets of tissue and then paste it back onto the person. But nevertheless, this type of tissue, which is derived from the patient and then stuck back onto the other parts of the patient, this is known as an autologous graft. Or an autograft. Then, if you have a graft from someone that you are genetically related to, either a twin sibling or a clone, then your genetic phenotype is essentially identical, or very as close as you can get. And so this has the highest chance of being accepted. And this is known as a syngeneic graft or an isograft. If you, because of the, this is probably the most common type of graft, because of the difficulties in getting genetically identical matches, we normally just, in desperation, you take grafts from anyone off the street. And generally with immunosuppressive now, graft rejection is not that much of a problem. But when you have this big difference in the genetics between the donor and the recipient, this is known as an allogeneic graft or an allograft. Going back to this problem of finding enough donor organs, there's a lot of experimentation, and in fact, just a couple of days ago, it was announced that the first successful graft has been done in this type. And this is where you take an organ from another species, in particular pigs, because their organs are around the same size as the human organs. And so these are xenografts or xenogeneic type of transplant. So in the early days, this is immediately after the Second World War, a lot of doctors were doing, and surgeons were doing research based on the success of blood transplantation, blood transfusions. Doctors were researching ways of possibly taking parts of body parts from a person and putting it into a recipient. And this is because of the horrific injuries that many of these soldiers suffered from the war. And from another stream, there were pure immunologists, not clinicians, who were looking at ways of transplanting bits of tissue from one mouse to to another. The problem in the human system was the fact that because we are such a genetically diverse species through all the recombinations of our ancestors in terms of our genes that come up to us as individuals, the trying to figure out what was happening wasn't possible, and still not possible. So what the immunologists did was they went into collaboration with animal breeders to generate identical clones of mice, identical species of mice. What they do is they mate a male and a female mouse, and from the progeny they make the brother and sister. That progeny, they make the brother and sister, and they do this over 50 generations. Basically what you're doing is diluting out the genotype of these animals so that eventually you get what are known as purebred strains of mice. And this was done around about the 50s, and these mice are still the workhorse animal models for immunological experimentation and all medical experiments now. So these strains of mice are essentially like clones that all members of that species have around about 95% similar genetic identity. You can't do this with humans. And with having established these purebred strains of mice, they could then take out bits of tissue, particularly skin, from one strain and put it into or graft it onto the skin of a recipient mouse. So in this experiment here, you can see that you cut out a bit of skin from this green mouse and you transplant it onto the recipient purple mouse. And if the genetic relatedness is very different, after 10 days, that graft will start to necrose. It's not getting enough blood, and it will die off and slough off the recipient. If then they took lymphocytes or blood from this mouse that had rejected that green skin and injected those blood cells into another purple mouse, and then did the same experiment where they transplanted green skin onto this immunized mouse, instead of getting rejection after 10 days, you get rejection after 3 days. So the conclusion from that was that in this initial transplant, you had immune cells that had been generated in this mouse that rejected this green skin, and you were transferring these immune cells into the second purple mouse, such that when it got that transplant, those transferred immune cells could immediately kick off to kill that green graft. The other clue was that in this original purple mouse that received that green skin and rejected it, if they let that mouse rest for 6 months or so and then repeated the experiment, grafting the green skin onto that original mouse, instead of rejection happening after 10 days like it did in the first experiment, Now this mouse rejects it after 3 days, showing that having received that initial green skin and then rejected it, you had memory cells that had been established in this mouse, such that when they got a repeat exposure to this green skin, these memory cells could immediately kick in. And then it was also shown that this type of immune response is specific, because in this experiment where the second purple mouse received these immune cells from that initial purple mouse and rejected the green skin, if that mouse got a skin graft from a third donor, not the green skin, rejection happened again like a primary response after 10 days. So it means that those transferred immune cells were specific for the green skin, but not for this third graft. So all of this behaving like a classical immune response that we would expect. And then, as we covered this before, we find that we can, the immunologists developed these genetically modified mice known as nude mice, which don't have a thymus. So they don't have a thymus, that means they don't have any functional, what? T lymphocytes. And without functional T cell response, when they do these type of transferences of skin from xenogenetic donors, such as humans, cats, chickens, chameleons, blah, 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 onto these nude mice, you find that rejection happens a lot more slowly compared to these wild-type mice where rejection can happen after 3 days. So this shows that the immune mediators of this rejection response are predominantly T lymphocytes, T cells. So based on that, the only ways of trying to match the donor and the recipient was to do this type of experiment known as the mixed lymphocyte reaction, where you culture immune leukocytes from both the donor and the recipient. But before you do that, what you do is you make the recipient cells targets. So basically you treat the recipient cells with high energy X-rays. And what that high energy does is to break up the DNA of these cells such that it's not enough to kill the cells, but the cells can't divide. So they're viable, but they're stationary. And so basically, then you are creating a target cell. So when you mix the non-irradiated donor cells with the irradiated recipient cells, if they are unrelated, if there's no match, the donor's CD4 and CD8 cells will attack the recipient's stationary cells, either in the form of cytokine production, which you can measure, or direct killing from the donor CD8 cells onto the recipient's cells, such that you will see increased death, cell death. If there is more genetic relatedness, this reaction doesn't happen, and you don't see increased lysis or increased cytokine production. Right? So this is the basis of the mixed lymphocyte reaction. from those single bred colonies, those pure strains of mice, and from those experiments that I talked about of grafting a bit of skin from one strain onto another, they established these so-called laws of transplantation. And so in this first case here, which resembles an isograft, what they do is they took skin from one strain of mouse, strain A, and transplanted it onto another mouse of the same strain, also strain A. And you find that in that kind of transplant, there is no rejection. So basically, this is like a clone of a mouse being transplanted with skin from identical genotype. Yes?
Same strain meaning same genome?
Yeah, same genome. All right? In the second experiment, they take skin from a different strain, strain B, and graft it onto our original strain A mouse, and you get rejection, because strain B and strain A are genotypically different. And so the recipient's immune response is then attacking this grafted skin from strain B. If you mate strain A and strain B together, so then your hybrid has molecules from both the strain A and strain B. And then you do this experiment where you take the parental strain skin from the strain B and put it into this hybrid strain. Because there are identical or similar proteins from this B and this hybrid, there is no reaction. Because this grafted skin, there is no difference, there's no change. Your question?
Yeah, and the third one, the one you're talking about right now, why wouldn't the green part attack the orange part?
Because this progeny, this hybrid, contains both A and B elements, right? But you are transplanting only B elements into this mouse. So even though there is no A involved, this is not seeing any other foreign type of element. But the opposite is not true, because here you transplant skin from the hybrid AB and you put it into this recipient that is only strain A. So you are introducing B elements into this A, which it will see as foreign and attack, and so you get rejection. Okay? So then with the development of molecular biology, we understood that these elements which make up whether this is strain A or strain B is determined by the MHC or HLA antigens. And as you remember, the importance of these HLA antigens or MHC antigens is that they process and present self-proteins or self-peptides onto the surface molecules to be recognized by the T lymphocytes. So just to revise what I told you about last year, we have two classes of HLA molecules. Class 1 is A, B, and C. Class 2 is DP, DQ, and DR. The characteristics of these HLA antigens is that they are codominant. That means that you express 6 species of class 1 and 6 species of class 2, both from your mother and your father. So in terms of tissue typing techniques, for real-time or real-world tissue transplantation, particularly in renal transplants, they don't worry about matching all three classes of all three alleles of class 1 or all three alleles of class 2. They just match for class 1 A and B and class 2 HLA-DR. I guess because these, just based on immunology, these are the most prominent targets or dominant antigens recognized in this process. The mismatches in the others can be diluted out with using immunosuppressives or whatever. So with genotyping, what you're essentially doing is looking for the least difference between the donor and the recipient. So if you take this example here, we have 5 potential donors, and we are looking at A class, HLA class 1A, B, and class 2DR. These two numbers denote the paternal and the maternal genotypes, right? So our recipient in HLA-A has the alleles 3/8, in B has 1/8, and in D 3/4. Now, at this start, it doesn't matter whether this is denoted as 1/8 or 8/1, because there's an expression on each cell in the body that is expressed of the mother and the father's HLA, right? So what we want to do is to look at these donors, and we want to see the closest match between the donor and the recipient. So we look at the first case, the donor 1. We see that 3/8 and 3/8 here is identical. So there's no difference there. 1 is the same here in class B, but there's a difference there, right? Mm-hmm. So that's one mismatch. And then this one here, there's another mismatch between the 8 and the 3, because the 4 is identical. Yeah? So the conclusion from that is that there are 2 mismatches with donor 1. So with donor 2, how many mismatches would you see?
3.
There's one mismatch here.
Oh, this is a mismatch.
5.
Oh, one of them.
Right?
2. There is
2.
There's one mismatch here. There are 2 mismatches here. And 2 mismatches here, which makes a total of 5. Okay? So donor 3?
3 would be 1, 4, 3.
3.
You're looking at the one.
Donor 4?
4.
And donor 5?
1.
3 or 2?
1.
Where do you get 3 or 2 from? Because remember, just like that figure with the mice, with the rules of transplantation, you got 3/8 and 3/3, right? There's no difference there. There's one difference there, and it doesn't matter, like I said, whether the numbers are in sequence. So 3 and 4 is identical to 4 and 3. Okay? So here the conclusion then is that donor number 5 is the ideal donor.
So in A there's no mismatch.
Compared to the recipient and the donor, no, there's no difference, because even though recipient has allele A, has allele 8, the donor is identical in terms of being homozygous for 3. Alright? So that's one of the ways of measuring it. The other method, which is similar to the mixed lymphocyte reaction, which I talked about in this slide, is that instead of looking for cytotoxicity or cytokine production, what happens is that when you get an activated immune response as a result of recognizing these differences in the HLA, your reactive immune cells will proliferate. And you can measure this proliferation by adding radioactive thymidine so that it gets incorporated into the DNA. And so just by measuring radioactivity in these cells, you can measure proliferation. So this is another type of mixed lymphocyte reaction where you again irradiate your target cells from the recipient. And you add, this is the way, you irradiate the target cells from your recipient, and you're adding your donor cells to that. And if there is a, if they are closely MHC matched, you don't get any proliferation, so you don't get any incorporation of radioactivity. So you're looking for those samples which have the least radioactivity in them. And so you generate a graph like this where you have different numbers of active donor cells added to irradiated recipient cells. In this case, 100 donor cells to one recipient, 1,000 donor cells to one recipient, and so on. So obviously, the more active responsive cells you have, the more proliferation you're going to get if there's a big mismatch, right? So in this first case here, where we've got allogeneic dendritic leukocytes, and in this allogeneic response, you can see that the more of these leukocytes you add, the more proliferation you're going to get. Here you have an essentially autologous reaction where you're adding the recipient's own cells, which are active, to the recipient's irradiated cells. There is no reaction. They are not going to proliferate because they are recognizing identical MHCs. So you get low counts here. This y-axis represents the amount of radioactivity in the culture medium. And then in these controls are just the irradiated cells alone or the recipient cells alone. No difference there. So again, the more change you see, or the highest increase in radioactivity means that it's not an ideal match because there is a reaction. Okay? So then from that, you do this proliferation assay where you have your recipient cells mixed with either of these three potential donors. Yes.
Then why is the yellow line even elevated if there's no response? Why isn't it flat like the blue one?
A lesser degree of mismatch.
You told us there's no response.
Autologous.
Yeah, because there's nothing to react against those target cells.
Then why is there elevated white blood cells? Why isn't it flat like the blue one?
Why? Sorry,
Why is the yellow one elevated basically?
a slight
Why isn't it flat?
increase.
Yeah.
That's just a technical one of the technical artifacts of this thing. Nothing is absolute. So even though you radiate these cells, you don't essentially block proliferation in 100% of these cells. Okay, so it's just minor. So in this type of checkerboard titration, here you have your recipient cells irradiated, and you add non-irradiated donor cells, and you're looking for the least value, which will tell you the least proliferation and therefore the least reactivity. So here, just as a control, we're adding the recipient cells active to the recipient's own cells irradiated as targets, and you get a baseline value of 1. If you add the father's cells to the recipient's irradiated targets, you see a proliferation index of around 4. The mother has an index of around 2, so she's not as reactive as the father. And the sibling has 1. So what does that tell you? What's the best donor? The sibling, right? So this is basically the essence of this type of test. Now, in terms of the immune reaction, the reactivity of an allogeneic response is 1,000 to 1,000,000 times higher actively in terms of cytokine reduction, in terms of cytotoxicity, compared to when you have a natural infection by a bacteria or a virus or so on. And there's several reasons for this. One is that your antigen load is much higher when you are trying to transplant a kidney or a liver into a recipient. Because imagine that transplant, those are all non-self antigens, much more than if you get a natural bacterial replication or virus replication or whatever, right? That is one point. The other is that the immune reactivity in this rejection, the reactive T cells, which normally undergo thymic education, processed and deleted from reacting against self-MHC, they have not been primed not to react to the MHC that is being given in this donor organ. Because biology was never meant to take bits from another person to put it into our bodies. So this is totally artificial. So these T cells, which are reactive to this foreign tissue, have never been removed not to react to these tissues through thymic education. Right. Another thing is that a large part of these donor-specific T cells have actually been generated to act against other types of antigens like bacteria or virus infections. And so just by chance, their specificity of their T cell receptors are cross-reacting with this foreign MHC self-peptides expressed on these antigens. So antigen load and also the quality of these foreign antigens as brought in by this transplant makes this a much more robust or most severe type of reaction compared to a natural infection. As an example, in a normal response to a bacterial infection, you find that 1 in a million or 1 in 10 million cells, lymphocytes, are specific for that bacterial virus. But in terms of a transplant rejection, 1 in 10 or 1 in 100 of these immune cells are attacking this donor organ. So the degree of damage as a result of the immune response is much more severe. And the target of that immune response is generally against the blood vessels, the endothelium. And by attacking the endothelium of the transplant, you are preventing normal blood supply to the transplant, and so the transplant's not getting enough oxygen, and it's going to necrose and die after so many hours or whatever. So, but with modern immunosuppressives, even though because of this problem of getting close matches between donors and recipients, nevertheless the advances in immunosuppressive drugs have helped to reduce or negate the need to closely match HLAs. You can get away with this. The big difference is in terms of bone marrow transplantation, where you need strong matches, because if you don't, you can get graft versus host disease. The other thing is that even if you have a twin brother or sister who you can bribe to give you a bit of her kidney, there is still a chance that you can reject this organ, this transplant after a few years, because even though you are very close genetically, there are still minor differences, such as the fact that the say a female sibling has X antigens that your male Y genes don't have, that can see as foreign. These are known as minor histocompatibility antigens. Another factor which determines the chances of the transplant being accepted or rejected is how well, in what condition the transplant organ is before it's given to the person. So obviously, cadaver transplants generally don't work. If you take kidney or liver from someone who's been dead for more than 24 hours, that's not going to have a high chance of success. So the quicker you get from time of death into the recipient, the more chance you have of it being accepted. The other, if you need to fly this donor organ from say a different part of the country to the hospital, 3 or 4 hours, the other factors you need to make sure is your preservation of that organ. It has to be cold, it has to be flushed with physiologically media to keep that organ as fresh as possible. Because if it doesn't, some of the cells that die can release DAMPs. And when they release DAMPs, that can stimulate an inflammatory response, cause upregulation of adhesion molecules, inflammatory cytokines, and all of that reducing the integrity of this donor. So because of the this upregulation of these DAMPs, then the innate immune responses can be activated, and then it bring in Th1, Th2 type, Th17 type inflammatory responses, which can then add to attack the donor organ. With regard to the types of rejection, we can have very fast, in terms of hyperacute rejection, which can happen within hours after transplant. The normal type of rejection which we associate is known as an acute rejection, which is type 3 and type 4 hypersensitivity response. And chronic rejection, which is probably the most common, because apparently even though people get successful transplants, whether they're heart, lung, or whatever, after about 5 to 10 years, most of these people need another transplant. So even though the transplant appears successful initially, over time there is low level of chronic rejection.
Even with immunosuppression?
Because of these minor histocompatibility complex antigens, or the reduction of immune suppression or whatever. And so with this chronic low-level damage of the transplant, then eventually you can get occlusion of the blood vessels and whatever, and so the organs no longer function. In hyperacute rejection, which as I said, is the rejection occurs after hours of rejection, this is in certain instances where the recipient has been exposed to different antigens, foreign antigens. This is in the case of mothers who've had multiple births, because every time they're pregnant, they are exposed to paternal antigens, so they can derive an immune response against these. So they develop these allogeneic antibodies. The same with people who've had multiple blood transfusions and people who've had unsuccessful transplants in the past. So they've been exposed to these foreign MACs. They develop anti-MAC antibodies. So when they get these transplants, these antibodies will immediately bind to these alloantigens, fix complement, and you can then get damage to the blood vessels and rejection. So this is a mixture of both type 2 and type 3 hypersensitivity. Acute rejection, as I said, is in a case of a mismatched transplant. This is a mixture of 2, 3, and 4 hypersensitivity. So both antibody mediated with complement damage to the endothelium, as well as cytotoxic T cells and inflammatory cytokines produced by Th2 cells. And in this, because you have all of these components, the rejection can occur after 2 weeks to a month or so. And then the chronic rejection, as I said, is due to these minor histocompatibility antigens. We have low level of immune attack, This is through a combination of both 3, 2, 3, and 4 type hypersensitivities. And because you have this damage, you get repair through TGF-beta. So over the years, you get occlusion of the blood vessels. This is just a repeat of what I've said, and that's it. Okay. Are there any questions?
Yeah, doctor, why did in the athymic mice it took them 60 days to fight the graft, even though they're athymic? Shouldn't that be normal?
So, well, nothing's absolute in biology. So even though they don't have any T cells, you can get other parts of the immune system like natural killer cells and so on also attacking the transplants.